everyone. In this session, we will start with the discussion of development of your nervous system. I hope the gross part is already finished in the general anatomy. So what are all the important structures you see in the nervous system? We can see the development of nervous system in the 4th week of intrauterine life, okay? We'll mainly see for the 3 important structures in the development of nervous system. The very first one is neural tube and the second one is neural crest cell. Neural crest cell, and the last one is, what is the last one? It is called ectodermal placodes. It is called ectodermal placodes. The formation of neural tube is called neurulation. The process is called as neurulation. Okay, so you should be aware of all the events or the development process that takes place from the 1st week to the 3rd week. Okay, now imagine by the end of 3rd week we already have 3 germ layers. We have ectoderm, endoderm and mesoderm. Okay, now I am going to remove the amniotic cavity part and I will see the structure from the above part. This is how exactly the structure will look. So this is the structure. We can see amniotic cavity, yolk sac, ectoderm and endoderm. In between we have intraembryonic mesoderm. Now I will remove this amniotic cavity part and I am seeing this structure from the above or from the top view. So this is how the structure will appear. So this is your ectoderm part. Okay, below this we have all the mesoderm and endoderm. So if I write the detailed picture of this ectoderm, this is how the structure will look. Okay, now in the detailed picture we have 2 ends. This is the cranial end and this is the caudal end. And all the dotted structure will represent your ectoderm part. So what are the important structures we see in the cranial and caudal end? What are the important structures we see in the cranial end and caudal end part? Okay. Okay. Okay, on the cranial end we have buccopharyngeal membrane part, and on the caudal end we can see the primitive streak part. Okay. So this is your buccopharyngeal membrane and this is your primitive streak. What is that structure present along with your primitive streak at the or by the end of 3rd week? What node? There is a structure called node. What is that node is called? we have one more important structure that is called Hansen's node. This is called Hansen's node. Okay. So between the buccopharyngeal membrane and the Hansen's node, we have a rod-like structure. This is the rod-like structure. This is called notochord. Okay. So now the ectoderm cells or the ectoderm part present along with your notochord will go under a differentiation. Once the differentiation takes place, this is how the structure will look. The blue part is your notochord, and this is the buccopharyngeal membrane, and this will be your Hansen's node part. So what is this differentiated part is called as? It is called as neural or neuroectoderm. And the remaining ectoderm at this level, it is called as surface ectoderm. So from this neural ectoderm, your entire nervous system is going to develop. It is the derivative. Okay. So the derivative of your neural ectoderm is the nervous system. What are the derivatives of surface ectoderm? What are all the structures that are going to develop from the surface ectoderm? What are the structures going to develop from your surface ectoderm? All the organs or structures that you see from your proctodium to stomodium is developed from your surface ectoderm part, that is from your mouth to anus part. Other than that, the epidermis, your hair and your nails are going to develop from your surface ectoderm, and the nervous system is going to develop from your neural ectoderm part. Okay. Any doubts in this? Any doubts in this figure? Okay. In this stage, your neural ectoderm is like a plate-like structure or a very flat structure. So, we name it as neural plate. We call it as neural plate. So, during the process of development, this neural plate is going to get folded by side to side direction and form a tube-like structure. This will develop like this. Okay. This tube-like structure is called as neural tube. Okay. This process of formation of neural tube is called neurulation. Okay. Any doubts in this? Any doubts? So once we start studying the detailed structure of your ectoderm, from the top view, we will get cranial end and caudal end. Between the cranial end and caudal end, we have buccopharyngeal membrane, notochord, and then Hansen's node followed by your primitive streak. Below the primitive streak, we can see the cloacal membrane. Okay. So from this structure, your ectoderm is going to differentiate along with your notochord part. Once the differentiation takes place, the ectoderm is divided into two parts. The first one is neural ectoderm, the second one is surface ectoderm. From the neural ectoderm, your nervous system is going to develop. In the initial stage, it is more like a plate-like structure, so we name it as neural plate. Later, it will go under a process of folding, forming a neural tube structure. Okay, is it clear for everyone? Any doubts? No doubts? Okay. So for the development of nervous system, we study the structure or the embryo in three aspects. Okay. The very first aspect is above view or from the top view, which I have explained just now. Other than that, we will study it in two other aspects. The second one is coronal section. The third one is sagittal section. So now we are done with the above view part. Next we will see the coronal section and sagittal section. Any doubts in this? Yes or no? Any doubts? Okay. So what are all the structures we can see in the coronal section of the embryo? During the folding of embryo, we have already studied the coronal section and sagittal section. So what are all the structures we can see in the coronal section? How to take the coronal section of embryo at least? How do we take the coronal section of embryo? So if this is an embryo, we are going to cut like this and we will see it from the front view. That is called the coronal section. So once we take the coronal section, this is how the structure will look. Now we will concentrate only on the ectoderm, mesoderm and endoderm part. Okay. I have already mentioned in the ectoderm part, the differentiation of cells will take place, forming a structure called neural plate. So I will mark the neural plate in red color. So this is the neural plate, or I can say it is the neural ectoderm. The remaining part is the surface ectoderm. We have notochord followed by endoderm. Okay. Now the cells in the intraembryonic mesoderm part will undergo differentiation and forms the folding of your surface ectoderm. The cells present in this region will undergo differentiation and causes folding in this particular points. Once the folding takes place, this is how the structure will appear. Okay. So now these foldings are named as neural folds. These foldings are named as neural folds on both the sides. So what are all the components of neural folds? It is already discussed in your general anatomy part of nervous system. What are the components of neural folds? Your neural folds have two important components. Those are neural crest cells and neural epithelial cells. This will be your MCQ question, so try to remember the names always. Your neural fold have two important components. The first one is neural crest cells and the another one is neural epithelial cells. Okay. So now the surface ectoderm part, the black part what you are seeing, will join together in the midline so that your neural plate is going to separate from the surface epithelium. This is your surface ectoderm and this is your neural tube. Okay. So we have seen the formation of neural folds. The component of this neural fold is neural crest cells and neural epithelial cells. So here the like cells will attach with each other. They both will start moving towards the midpoint and they will attach with each other so that your red part is going to separate from the surface ectoderm part. Okay, and here this part is called as neural groove. This is called as neural groove part. Okay, once this neural groove separate from the surface ectoderm, it is called as neural tube. Okay, so now we are left with surface ectoderm, neural tube, notochord and also the endoderm part. Any doubts in this? Any doubts? Okay. So now we all know the surface ectoderm have two types of cells. So the neural epithelial cells, this one, the second one is going to stay with your surface ectoderm only. So this layer what you are seeing is your surface ectoderm plus your neural epithelial cells part. The neural crest cell what you are seeing there, this one is going to come and present on both the sides of your neural tube. This is your neural crest cells now. Okay, this is how the formation of your neural tube, neural crest and also the neural epithelial cells takes place. Any doubts in this? Any doubts? Yes or no? Any doubts? Okay. This is how the section, the coronal section study will show the structures. Okay. In the coronal section, we have seen the neural fold, neural groove and also neural tube. These are the three important points you should remember: neural tube, neural groove and also neural tube. Okay. The last one is sagittal section. How we take the sagittal section and what are all the structures we see in the sagittal section? So what are all the structures we see in the sagittal section of the embryo? What structures we see in the sagittal section? If you answer something, we can continue. What structures we see in the sagittal section of the embryo? In general, I am asking. So what structures we see in the sagittal section of embryo? Any answers? If you both want to go out, you can go. Tell me what structures we see in the sagittal section of the embryo. How do we take the sagittal section at least? Okay. So what structures we see in the embryo? We are studying at the level of 4th week. So, what are all the structures that develop from starting 1st week to the 3rd week in the embryo? Elementary canal. Okay, fine. Neural tube. Okay. Neural tube formation takes place in the 4th week. Just before that, until the end of 3rd week, I am asking what structures you can see in the sagittal section. Any other answers? Any idea about somites? What are somites? How many pairs of somites are present? In which week or on particular which day we can see the somites? What are somites? Can anybody define somites? How many pairs of somites or how many number of somites present in the embryo? 2 pairs? 2 pairs. Okay, fine. Any other answers? How many pairs of somites? What is the significance of somites actually? Can we see somites in adults? No. Yes or no? Can we see somites in adults? No. Okay, so it is present only in the embryo stage. Okay, everyone just go and try to brush up the topic somites. It is very important. If you know the somites, understanding the sagittal section will be very easy, okay? So this is your surface ectoderm part. Okay, and whatever the blue structure you are seeing right here is your neural tube. If you observe carefully, your neural tube is in still contact with the surface ectoderm. It's because at this stage or at this level, your blood vessels are not fully developed. So, for the nutrient purpose and also for the nourishment of the developing neural tube, it will be in contact. It will be in contact with your amniotic cavity part. Amniotic cavity part. This opening is called as anterior neuropore, and this opening is called as posterior neuropore. Okay. So once the development of your blood vessels takes place, that is at which week? Development of blood vessels start or finish its development at which week? Remember the aortic arches development, when it will end, when it will start? Okay, once the development of your blood vessels takes place, your neural tube will not have contact with your amniotic cavity anymore. Okay, so the anterior neuropore and the posterior neuropore will close. Your anterior neuropore will close at which somoid level, and your posterior neuropore will close at which somoid level. So you should know what is somoids, what are the significance or importance of somoids, and how many pairs of somoids are present. Okay. Take this as an assignment question. Find out at which level of somoid or on which day your anterior neuropore and posterior neuropore will close. Okay. So once your anterior neuropore and your posterior neuropore will be no more contact with the amniotic cavity, it will look like a tube-like structure, just a hollow tube-like structure. Okay. The cranial end of this tube will undergo enlargement and the caudal end will undergo elongation process. Once the enlargement and elongation process takes place, this is how the structure will look. So this is nothing but the brain part. These are the vesicles of your brain, and the elongated part is going to develop as a spinal cord. This is your hindbrain, forebrain, midbrain and hindbrain vesicles. Okay. So by the end of your 4th week, the anterior neuropore will close at middle of the 4th week. Find out the somoid levels 4th week, and posterior neuropore will close by the end of 4th week. Okay. Once the closure of the neuropores takes place, this is how the neural tube will look. The cranial part will undergo in enlargement process and form 3 brain vesicles, that is your forebrain, midbrain and hindbrain, whereas the caudal part will undergo elongation to form your spinal cord part. So basically your neural tube will give rise to brain and spinal cord. Okay. These are all the important structures we see in the sagittal section study. Any doubts in this? Any doubts in the sagittal section? No doubts? Okay. Next, we have seen the neural tube which is formed by the process of neurulation is going to develop as your brain and spinal cord, that is the part of your central nervous system. Your neural crest cell is going to give rise to your peripheral nervous system. Okay. So remember, there are 2 neural crest cells that is present on either side of your neural tube. So in the initial stage, the neural tube will get separate. Beneath that we have notochord. So only your neural epithelial cells will get combined with the surface ectoderm part, and your neural crest cells will come and present here. Okay. Now this neural crest cell is going to divide into 2 parts and move in the dorsolateral aspect. Dorsolateral aspect. Okay. It will come here. Again, once they move to the dorsolateral aspect, it will again divided into 2 parts. These 2 parts are named as dorsal mass of neural crest cell and ventral mass of neural crest cells. Okay, the divided masses will present like this. So this is the dorsal mass and this is the ventral mass. Okay, remember your neural crest cells, once it gets separated from the surface ectoderm, it will be present in the middle, and here it will get separated into two masses, and first they will move in the dorsolateral aspect. Once they start moving in the dorsolateral aspect, it will again divided into two parts. Now we will name it as dorsal mass and ventral mass. These dorsal mass and ventral mass will present on either side of your neural tube structure. Okay, your dorsal mass will give rise to sensory ganglions and your ventral mass will give rise to autonomic ganglion. That is your sympathetic and parasympathetic ganglions. Okay, any doubts in this? Any doubts? Okay, so your neural crest cell will give rise to which nervous system? Peripheral nervous system, which includes sensory ganglions and autonomic ganglion. What happened to the ectodermal placodes? Your ectodermal placodes will give rise to hypophysis, that is your pituitary gland, and also to the inner parts or aspect of the ear. Okay, these are the events that takes place in the 4th week of the development of nervous system. So your nervous system will develop in the 4th week. It will start from the 4th week. By the end of 4th week, we will be having neural tube, neural crest cells and ectodermal placodes. Your neural tube formation process is called neurulation, and it will give rise to your central nervous system. So it will develop your brain and spinal cord. Your neural crest cells will develop or give rise to your peripheral nervous system. We can see the development of sensory ganglions and autonomic ganglions. The last one is ectodermal placodes. It will give rise to your hypophysis, that is pituitary gland, and also to your ear and few other structures. Okay, that is again one more class will be there. I will discuss it in detail. Any doubts in this? Any doubts? Yes or no? Any doubts? Oh. Nervous system is derived from the ectoderm, except its blood vessels and some neuroglial elements. The specific cell population of early ectoderm, which give rise to entire nervous system and special sense organs, is termed as neural ectoderm. The neural ectoderm later differentiates into 3 important structures. Those are neural tube, neural crest cells and ectodermal placodes. The neural tube gives rise to the central nervous system. The neural crest cells forms nearly entire peripheral nerve system. The ectodermal placodes will contribute in the cranial sensory ganglia, hypophysis and also the inner ear parts. Formation of neural tube. In early embryonic disc at about 16th day of embryonic life, the ectoderm overlaying the newly formed notochord thickens in the midline, forming the neural plate. Initially, the neural plate is flat and slipper shaped with narrow end toward the primitive node. Between the primitive node, we have which node? Hessen's node. Don't forget that, okay? The somatic mesoderm develops on either side of the notochord. The margins of neural plates are elevated to form the neural folds. As a result, center of the plate sinks, creating the neural groove. So you have to remember step by step. First we have neural plate. Once the neural plate formation takes place, we can see the formation of neural folds. Once the neural fold is done, we will see the neural groove. From the neural groove, we can see the formation of your neural tube. The neural folds gradually move together toward the midline and finally fuse to form a cylindrical neural tube that loses its connection with the surface ectoderm. The process of formation of neural tube is called neurulation. The fusion of neural folds begins in the middle, that is the region of the fourth somite on 20th day of embryonic development, and it is simultaneously proceeds in the cephalic and caudal directions. The fusion at the cranial and caudal ends of neural tube is somewhat delayed, forming small openings called anterior and posterior neuropores. The neural tube and overlaying amniotic cavity therefore remain temporarily in open connection with each other until the development of your blood vessels takes place. Okay. The entire neuropore closes earlier in the middle of the fourth week, as I already mentioned, at the week 18 to 20 somite stages. So figure out what is somite stages means and also the significance of somites in the developing embryo part. Okay. And the posterior neuropore will close by the end of the fourth week. By the time the neural tube is completely closed, it is divided into an enlarged cranial part and elongated caudal part, which later on give rise to your brain and spinal cord respectively. This is the picture what I have explained. Take 2 minutes and see the structures once. First you can see the formation of neural plate, followed by neural folds and followed by neural groove. At last we have the neural tube. On either side of the neural tube you can see the neural crest cells. So when you see the neural crest cell, you should be aware that it is formed of two masses, dorsal mass and ventral mass. Any doubts? Any doubts in this picture? No doubts? Formation of neural crest cells. As the neural folds come together and fuse, cells at the tip of the neural folds break away from the neuroectoderm to form the neural crest cells. The surface ectoderm of one side becomes continuous with the surface ectoderm of the opposite side. Thus, cells at the tips of the neural folds do not participate in the neural tube formation. The neural crest cells at first remains in the midline between the dorsal surface of the neural tube and the surface ectoderm, and then it forms two clusters, that is dorsolateral. First it will move in the dorsolateral aspect, and later that dorsolateral aspect mass will divide into dorsal mass and ventral mass of your neural crest cells. The neural crest cell differentiate to form the dorsal root ganglion, sensory ganglion of your cranial nerves, and also the melanocytes, chromaffin tissues and some other cells of your nervous system. So these are the derivatives of your dorsal mass. It forms the dorsal root ganglion of spinal nerves and sensory ganglion of the cranial nerve 5, 7, 9 and 10. It also forms few cells, that is neuroblast cells, pluripotent cells and spongioblast cells. Okay. Ventral mass cells will give rise to sympathoblast cells, that is it will later forms your sympathetic chain part, followed by parasympathetic ganglion and chromaffin cells. So what is chromaffin cells? What is chromaffin cells? First, this structure is actually called as sympathetochromaffin organs. Later it will divide into two parts, sympathetic cells and chromaffin cells. So what do you mean by chromaffin cells? So this is the structure of the formation of your neural crest cells. You can see this is the first mass. Okay. So here this mass is going to develop divide as a dorsal mass and ventral mass. From this dorsal and ventral mass, all your cells of the nervous system is going to develop. Any doubts in this? Any doubts in this diagram? No doubts? Whatever the theory I have written there as sensory ganglion and autonomic ganglion, here they have mentioned each and every cells that is going to derive from your neural crest cells, okay. But mainly you should remember dorsal mass is going to give rise to your sensory ganglion part and ventral mass is going to give rise to your autonomic ganglion part. From this part we are going to learn how the development of your brain and spinal cord will take place. Okay, stop talking. Any doubts in this chapter? This will finish your development of nervous system. So any doubts? No doubts? Okay.